We as humans are evidently tailless, the only evidence remaining that we once did being through the Coccyx, a small remnant of our vestigial tails that through our evolution became less and less advantageous as our ancestors became more terrestrial. The specifics of said loss, long since remaining unknown, have been covered in a recent paper that has unearthed that a simple genetic change led to how present our tails are today. That's been through an itinerant or travelling piece of DNA that's moved to a new chromosomal region which fundamentally changed how great apes formed a key developmental protein. Primate genome sequencing projects have made possible the identification of causal links between the phenotypic and genotypic changes in these animals, as well as enabling and assisting in the search for hominoid-specific genetic elements which control tail development, and this study is one key example of this. The paper, while currently being a preprint, presents the evidence that tail loss was mediated through the insertion of an individual allele element into the genome of our ancestors, which was found through the search of any ape-specific changes in the tail developmental genes. A gene known as TBXT was identified as a strong subject, as it contained a short DNA insertion established earlier as an allele element, which is present in all great apes, but missing in other primates. It was found that the allele sequences can move around the genome, thus giving them the name of jumping genes or transposable elements. Possibly the remnants of ancient viruses, allele elements are common throughout the human genome, making up around 10% of our DNA. Sometimes said insertions interrupt a gene and prevent its protein production, as well as in some cases changing where or how a protein is expressed, which makes them a huge driver of evolutionary variation. The previously mentioned TBXT gene codes for a protein known as Erasure, Greek for short tail, as said mutations can lead to mice born with shorter tails, as noted in previous experiments. At first glance, however, the ape-specific allele element did not seem to cause any significant disruption in the gene. On closer inspection, however, researchers did notice a second allele element nearby the first, with this element being present in monkeys as well as apes, with it being realised that in apes at least, the two alleles could stick together, forming a loop that would alter the TBXT expression so that the resulting protein would be shorter than the original, leading to a hominoid-specific splicing event. It was indeed found that human embryonic stem cells make two versions of the TBXT messenger RNA, one longer and one shorter. To study the effects of this splicing event, a mouse model was generated that mimics the expression of the human TBXT products by expressing both full-length and exon-skipped isoforms of the mouse TBXT. Mice with this genotype did indeed exhibit the complete absence of a tail or a shortened one, supporting the notion held that the exon-skipped transcript is sufficient enough to induce a tail loss phenotype, albeit with incomplete penetrance, the extent to which a gene is expressed. This meant that as the genetically altered mice had a mix of tail lengths, other genes must also be working together to eliminate tail development altogether in apes and humans, which are still unknown of. Although the ape-specific allele insertion was indeed a critical event when great apes diverged from other simians around 20 million years ago. It was also noted that mice homozygous for the exon-skipped genes exhibited embryonic spinal cord malformations, resembling a neural tube defect condition which affects 1 in 1,000 human neonates, which can in some cases produce spina bifida. It was therefore proposed that the selection over time for the loss of the tail along the hominoid lineage was associated with an adaptive cost of defects, an evolutionary trade-off that still echoes across time and continues to affect human health today. All in all, I thank you for watching this video, and that you may have learned something new. If you would like to see more from this channel, be sure to subscribe if you haven't already. And with that, I'll see you next time, whenever that may be.